So yeah, great to have Georgia with us. Georgia is currently a PhD candidate at Johns Hopkins Bloomberg School of Public Health, studying in the Department of Molecular Microbiology and Immunology, and she did her undergraduate training in microbiology and immunology from McGill University in Montreal, Quebec. So yeah, great to have you with us. And you're going to be speaking today about optimizing pre-conjugation of biotinylated MHC class I pentamers to streptavidin-fluorophores for simultaneous analysis of different antigen-specific T cells. So yeah, over to you, Georgia. We're looking forward to hearing your talk.
All right, sorry for the mouthful of a title. Thank you so much to ProImmune for inviting me to speak today about some of the work we've done with their MHC class I pentamers to study CD8 specific T cells in different viral infections. However, today I'm going to mostly be speaking about HCV. So I just wanted to do a quick introduction to hepatitis C virus, HCV, and just some important factors about HCV that kind of led us down this optimization pathway. So one of the most important things to know about HCV is that it's a highly variable virus. So it's highly variable on a superficial level in that there's like 6 major genotypes and multiple subtypes. But even in a single infected patient, there is a lot of variability in HCV sequence due to its high replication rate and lack of proofreading activity by its RNA-dependent RNA polymerase. So I just have an image here of what the HCV genome looks like in the different regions and the different proteins it expresses, and I'll get a little more into that on the next slide. So not only is HCV highly variable, but what I'm showing you here are mapped CD8 specific T cell responses to HCV in a cohort of people at risk of HCV infection that we follow in the lab. So these are individuals who are at risk of infection due to injection drug use. And something that I'll just mention briefly, what's really unique about HCV is that it is a chronic viral infection. However, about 25% of people who acquire it can clear it. So that's the specific subpopulation of our cohort that we're interested in studying and phenotyping the T cell responses for. So these are individuals who can clear HCV, and in fact, these individuals have been followed for so long in the cohort, some of them for almost 2 decades, that we've been able to see them clear a primary infection and then clear subsequent infections. So what I've shown in these 2 graphs here is in this first graph, new responses per infection. On the y-axis, you have number of recognized T cell epitopes. These are the mapped CD8 specific epitopes or MHC class I epitopes, and then their different infections on the x-axis. So what I really want to point out is you can see each symbol and color represents one individual in this subgroup of our cohort. And for some of them, they, in a single infection, can recognize up to 15 new MHC class I epitopes, and that's not including like maintained epitopes from a previous infection. In this next graph, responses per region, I have just the different regions of the HCV genome that I showed in the previous slide on the x-axis, and on the y-axis, again, you have number of recognized peptides or MHC class I epitopes. And again, every symbol and color is a different participant. Or responses from a different participant. And the take-home message from this graph here is just that not only do these individuals recognize multiple HCV epitopes in a given infection, those epitopes are also pretty uniformly distributed across the HCV genome. So there's not like a single immunodominant area where if we just kind of look at responses to that region, we'll be capturing the full response to HCV. And then the last thing that I'd like to say about HCV when it comes to studying the T cell response to it is this is actually a paper from my PI's lab, Andrea Cox, from 2008, where they looked at HCV-specific CD8 positive T cells and PD-1 expression. And really what's important is that they showed when there is no sequence evolution in the epitope that T cell recognizes, PD-1 MFI stays the same. However, if there is sequence evolution, you do see for a given HCV infection, some antigen-specific T cells will actually decrease PD-1 MFI. So this all makes sense. If the TCR is not being engaged because the epitope has mutated, you obviously wouldn't see your typical activation markers like acute PD-1 expression or CTLA-4, GLUT1. However, this is important for our study because if we do see a phenotype of T cells associated with an infection state in someone infected with HCV, we really want to be able to distinguish whether or not that's due to the fact that the epitope has undergone sequence evolution or if it's recognizing an intact epitope and actually doing something important. So all that to say is really what we were trying to do in our study is using MHC class 1 pentamer staining for a cohort with diverse T cell responses. And in addition to this pentamer staining and pulling out these HCV-specific CD8 positive T cells, we're using spectral flow and a high-dimensional flow cytometry panel to phenotype them. So this is just looking at the spectral emission of our panel and the different fluorophores. What is really critical here is we're at 27 fluorophores in this panel, so it's really hard to add new fluorophores. And so what we wanted was to kind of designate 2 fluorophores in this panel. In this case, we picked APC and PE as pentamer fluorophores. So each sample that we stained gets stained with the same grouping of fluorophores, and the pentamers are always on APC and PE. However, due to the reasons that I just mentioned in the previous slides, we wanted different epitopes to be stained by APC and PE, and we wanted to be able to distinguish those epitopes as unique CD8 positive T cells recognizing HCV. So we wanted to be able to use biotinylated MHC class 1 pentamers from ProImmune and streptavidin-fluor, but be able to distinguish which pentamer was on APC and which pentamer was on PE after staining our cells. So that's going into really the 2 parts of my talk for today. Is at first we asked, can we pre-conjugate biotinylated MHC class 1 pentamers to streptavidin-fluor to stain antigen-specific responses? And then we wanted to know if we could do that pre-conjugation, conjugate 2 different pentamers to 2 different fluorophores, stain in the same sample, but still pull out 2 unique epitope-specific CD8 positive T cells. So just really briefly, normally what you would do, right, is you would take your biotinylated pentamers, add it to your PBMCs during your surface stain, and then throw in your streptavidin-fluor during surface. So what we really wanted to do was take our biotinylated MHC class 1 pentamers from ProImmune, pre-conjugate them to our streptavidin-fluor, and then take that pre-conjugation and add that to our pentamers to be able to pull out our antigen-specific cells. So this is what I mean when I'm referring to pre-conjugation. We titrated 2 different aspects of this. So first we titrated how much streptavidin-fluor to add to our biotinylated pentamer to do the pre-conjugation, and then we titrated the volume of that pre-conjugated pentamer fluorophore to add to our PBMCs to pull out our antigen-specific CD8 positive T cells. And we did a lot of this optimization just using healthy donors to start with, just to see if it would work. So in the next couple of slides that I'll show you, we were looking at a healthy leukopak donor that recognized immunodominant influenza MHC class 1 epitope, as well as an immunodominant CMV MHC class 1 epitope. And again, just to reiterate, we titrated that ratio of pentamer to streptavidin-fluor that we did the pre-conjugation with, and then we titrated how much of that pre-conjugation we added to PBMCs. So this is just briefly what we stained these optimization experiments with. So we just used a smaller panel. And in these experiments here, we're just looking at a single pentamer stain. So in following slides, I'll show you what it looks like with 2 different pentamers on 2 different fluorophores. Briefly, our gating strategy. So we originally looked to do this with FITC, PE, and APC, but we moved forward with APC and PE for our final panel. So this is FITC on that CMV immunodominant MHC class 1 epitope. And this is just a legend of how to read this figure. So what you have, if you move horizontally along this figure, along these panels, that we are decreasing the ratio of streptavidin-fluor to pentamer. If you move down vertically, that's decreasing the volume of that pre-conjugation that we added per well. So what was very clear from these original experiments was that it was really important to actually titrate both aspects of this pre-conjugation. And from here, we could kind of pick a ratio of streptavidin-fluor to pentamer that we thought looked really nice and that we pulled out an antigen-specific population and reduced background. I'll also show you for PE on the immunodominant influenza MHC class 1 epitope. So again, moving vertically, you are decreasing the ratio of streptavidin-fluor added to the pentamer, and moving down horizontally is decreasing the volume of that pre-conjugation added to the PBMCs. And here as well, PE seemed to have a bit of a different titration, but still we were able to pick one that we felt reduced background, but still pulled out our antigen-specific cells. The next thing that we wanted to make sure was that these titrations that we were choosing weren't epitope specific, because obviously in the Hep C cohort, where we want to use these methods, there's a lot of different epitopes recognized, and this would be very inefficient if we had to change the titration of fluorophore depending on the epitope that was loaded onto the pentamer. So what we did here was we repeated this experiment using just PE, and we used we put PE on the flu immunodominant epitope and the CMV immunodominant epitope, both on PE, stained in different wells, and just wanted to see if we could use the same titration for 2 different epitopes to pull out a clean antigen-specific CD8 positive T cell cloud. And you can see that even though we're staining with 2 different epitopes, here flu, here CMV, the same titrations actually pull out the same fluorophore titrations pull out nice, clean CD8 positive antigen-specific little clouds. So then we wanted to see if we could do this in combination. So it's essentially the same setup, whereas this time we were in the same well staining for both the flu epitope and the CMV epitope, but on different fluorophores to see if we could pull out flu-specific CD8 positive T cells and CMV specific CD8 positive T cells doing this pre-conjugation method. So it was essentially the same thing, except this time we are adding a second pentamer on a different fluorophore. And so I'll just go over these top panels here for the sake of time, since the bottom is essentially the same thing, but with PE. So we can see that on APC, we have our flu-specific epitope. Oops. Our flu-specific epitope. On FIT-C, we have our CMV-specific epitope. And then if we gate on our CD8 positive T cells and look at APC versus FIT-C, we can see that those are 2 discrete populations. So our pre-conjugation can pull out 2 discrete populations in the same well. And then lastly, in the last couple of minutes, I just want to go over a quick pilot experiment that we did where we actually were able to test this method in PBMCs from individuals who have acquired HCV virus or HCV. And we picked PBMCs from 2 participants who went on to develop persistent HCV infection and 2 participants who went on to clear their initial HCV infection, and did a very similar setup where we have each PBMC sample was stained with 1 MHC class 1 pentamer specific for a specific HCV epitope on PE and another for a different epitope on APC. And then in addition to these MHC class 1 pentamers for HCV, they were also stained with our 27 color phenotyping panel. So this was kind of putting everything together, seeing how the pre-conjugation would look in addition to our phenotyping panel. So this is the workflow here where we have, we stain with 2 different HCV-specific MHC class 1 pentamers. We do our surface stain and intracellular stain with our 27 color flow panel, so then we can get phenotypically distinct antigen-specific cells. Okay, so this is some representative pentamer staining from that pilot study where we can see this is for a single participant where they had 1 HCV epitope stained on APC and another HCV epitope stained on PE, and you can see we get CD8 specific cells with very little background for both of these epitopes. And then when we gate on the CD8 cells and do PE versus APC, those are 2 discrete populations. Again, with our pre-conjugation, we were able to do this in a cohort of PBMCs from individuals who have HCV. And then from there, the last slide I just wanted to show was we are actually able to phenotype these HCV-specific pentamer positive CD8 positive T cells. So in red and orange are HCV-specific T cells from individuals who cleared their initial HCV infection, and in blue and green are the HCV-specific CD8 positive T cells from individuals who went on to have persistent HCV infection. And what was really nice about this is each of these populations, so pentamer 1, pentamer 2, those are 2 unique two unique epitopes, pentamers for two unique HCV epitopes on either APC or PE. And what we can see is that the expression of these markers really correlates more with viral outcome than with, for example, what fluorophore that pentamer was conjugated to. So this was kind of a nice way of tying it all together, showing that we were able to titrate streptavidin-fluor to our biotinylated pentamer in an appropriate way to pull out unique populations of antigen-specific cells within a single well. And then we could actually differentiate those T cells, recognizing those unique epitopes. And when we go to look at surface expression of markers of interest, those markers actually do seem to correlate more with infection outcome than with the whatever fluorophore the pentamers were pre-conjugated to. Another couple of interesting things here is historically this has been shown. Some of these markers have been shown to follow these patterns in individuals who clear HCV versus persist, particularly CD127. Our collaborators have published almost like a decade ago that these are the types of patterns of CD127 expression that you see on HCV-specific CD8 positive T cells in cleared infection versus chronic infection. So that was also really nice that with this panel and our pre-conjugation method, we are actually seeing similar trends and results to what has historically been seen in the literature as a validation. So to end, I'd just like to thank my PI, Andrea Cox, who's been very supportive in all this optimization and allowing me to do this, as well as Dr. Kimberly Russo, who started all of this with me and has recently graduated, Dr. Elizabeth Thompson, who's helped us with all the flow cytometry here, as well as other grad students in the lab, and Dr. Justin Bailey, who's provided mentorship, and lastly, the Lauer group from MGH, who has been very, very helpful and provided a lot of feedback. So thank you, and be happy to take any questions.
Great, thanks, Georgia. That was a lot of great flow data. Nice to see that. Are there any questions for Georgia? Anyone? Okay. Oh, yep. Jeremy, please go ahead.
Thanks, Georgia. Yeah, I have a quick question just about the overall principle, because obviously the biotinylated pentamers are kind of like one option of being able to then tag any color that you like onto this, and then also there's the unlabeled pentamer option, which you can then use in the first place to kind of then put these different colors on. So I'm just trying to understand like the overall objective in regards to when you stain with the for example, you're pre-conjugating the biotinylated pentamers. I may have just missed it, and I'm just trying to get my head around it a little bit because we're thinking about these all the time. But so you've obviously got the you've got your pre-conjugating your pentamer to the PE, and you're pre-conjugating another one to the APC. Is that just because you wanted the flexibility of having one buying one pentamer and having the different options? Is that
yeah, it was really just about flexibility. So one option was to just buy. So the other difficulty is each participant recognizes a different combination of HCV epitopes. So we wanted the flexibility of being able to say, oh, for this participant, I'll just pre-conjugate these two epitopes to PE and APC, but for another participant, take one of those epitopes and put it on like a different fluorophore than for another. So that was really what it was about.
Yeah, no, that makes sense. So I guess the way that we would normally kind of approach the challenge would be to stain the cells with the unlabeled pentamer, and then come in with the fluoro tag, which basically binds onto the end of the epitope tag that's already pre-labeled with APC or PE.
Yes.
So then you would just add that into all your other flow panel. Was the problem that couldn't be used in conjunction with your multicolor flow panel?
The problem there was then I needed to be able, if I'm understanding your question, I needed to be able to know what epitope went on which fluorophore. So I needed to know let's say in the flu case, I needed to know like that the flu epitope went to APC and the CMV epitope went to PE. So if I was doing the traditional way of doing the biotinylated pentamer staining, sorry, I feel like I talk about this a lot, so it makes a lot of sense to me, and then maybe not so much to everyone else, but
the pennies dropped. Pennies dropped.
Yeah, exactly. So I wouldn't be able to know what went where. So then if we're looking at the T cells phenotypes and trying to put it into context of sequence evolution, if I don't know what specific epitope was on APC, then I don't know what I'm looking at is due to sequence evolution or something weird about the virus at that specific epitope. So that's why we needed to do the pre-conjugation, so I would know what was on PE and what was on APC prior to staining the cells.
Makes perfect sense. Thank you for explaining it so clearly. That's great.
Thank
Thank
you.
you.
Great. Anyone else? Okay, well, thanks, Georgia.